session to welcome back Dr. Chopra. And he's going to give a talk that he alluded to about the microbiome. And one of the future things for us to look for is more information and understanding about the biome.
Thank you. I can get you some more water too. Perfect. Thanks so much. So this is, I believe, the hottest topic in medicine right now, microbiome. This is one talk that I have to revise virtually every week. There's something new. Then I have to decide, do I combine two slides? Do I take out one? Should I just put it in the summary section? And so on. So I'm calling it the second human genome, what shapes it, and how it actually shapes us. So I was invited by the Southern California GI Society 3 weeks ago at the annual meeting. And I gave a talk on leadership, and the second talk they chose was microbiome. And I said, why are you asking me to speak on the microbiome? You have a center at UCLA. And Erman Meyer, professor of medicine, is one of the experts. He's written a book, he has a blog. I said, this is akin to my bringing coals to Newcastle. It's an old British aphorism. However, I've mustered the courage, and I'm going to go ahead and give the talk. So this is the talk I gave there. I have nothing to disclose relevant to this talk. The concept of the human microbiome was first suggested by Joshua Lederberg. He was a Nobel laureate. Bacterial genetics is what he got the Nobel Prize in Medicine or Physiology. And he said the microbiome signifies the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. So 100 trillion bacteria in our GI tract. And then, of course, we have them in the nasal cavity, in the mouth, on the skin, other orifices. And if humans have 23,000 genes, the microbiome has 8 million genes. In aggregate, in the GI tract, they weigh 3 pounds. So it's been called the second human genome, the inner bacterial rainforest, and a newly discovered organ. And it made the cover of The Economist a couple of years ago. And the title was, Microbes Maketh Man. A word about the evolutionary timeline. Mammals evolved 200 million years ago, primates 60 million years ago, the genus Homo, including humans, about 2.5 million years ago. What about microbes? Appeared about, by best estimate, 3.5 billion years ago. So the GI tract is sterile at birth. It's colonized by bacteria within a few hours. And the bacteria that colonize are different if the newborn is born by C-section versus vaginal delivery. And then the microbial flora get established around 3 or 4 weeks. There's a little transition in some studies around 3 to 5 years of age. And more than 1,000 species of microbes take up house. I mentioned earlier, in aggregate, weigh about 3 pounds. And we're not talking about fungi, and not talking about viruses, and not talking about archaea, just the bacteria. Most of the research has been done on gut bacteria. So what shapes it? Whether we're born by vaginal delivery or C-section. There's some literature that kids born by C-section have a higher incidence in life of autoimmune disorders. Asthma, Crohn's disease, lupus, juvenile rheumatoid arthritis. There are 2 large-scale studies going on now in our country with actually randomizing newborns. And half of the newborns born by C-section, they will take a swab of the vagina of the mother within a few hours, within an hour of birth, and smear it on the face, in the mouth, on the skin. The other half will be a placebo, and they'll follow their bacteria and see what happens in terms of autoimmune disorders. Some obstetricians are already offering it to their patients and saying, we think this is a healthy way, you're going to get a C-section, do you think we can do this? And most women are opting for it. I'll come back to that. Whether we take antibiotics or probiotics, especially in the first 2 years of life, where we live, whether we live in San Francisco or rural Iowa, whether we travel, whether we have jet lag, what kind of diet, vegetarian, vegan, kimchi, do we eat fiber, do we drink coffee, whether we exercise. And some controversy in some studies, H2 blockers and PPI is shown to change the microbiome, in other studies, not sure to do so, and malnutrition. Now look at obesity, this huge epidemic in our country. Studies in mice as well as in humans show that gut microbiota differ in composition between obese and lean subjects. And there are notable differences in certain bacteria. So a very good study that got published in Science. And the reason it got published is they did a very clever part of the study. There are only 4 identical twins, sisters from Finland, where in each pair one is thin and the other is obese. Study the microbiome. The thin ones have a certain signature. The obese ones have a different profile. But the clever part of the study, they took the stool of the thin twins, gave it to genetically lean mice. They remained thin. They took the stool of the obese twin, mixed it with the food of the mice, genetically lean mice. They gained 30% body weight, developed insulin resistance. So obesity is associated with changes in the intestinal microbiota. The obese microbiome seems to be more efficient in harvesting energy from the diet. We all have patients who swear, Doc, I don't eat that much. I can't lose weight. We have to start believing them now. You can do calorie counts and maybe they're underestimating some of the calories they're taking in. So differences in gut microbiota composition might even function as early diagnostic markers for the development of type 2 diabetes. Butyrate, which is a product of intestinal microbes, may induce beneficial metabolic effects through enhancement of mitochondrial activity, prevention of metabolic endotoxemia, activation of intestinal gluconeogenesis. So again, most of the studies are small. Let me mention that at the start. Other studies are in mice, but this is a hot field. So 20 obese men with coronary artery disease randomized either to a Mediterranean diet or a low-fat, high-complex carbohydrate diet for 1 year. And they look at the bacterial composition, and it's altered. Both diets shown to exert a protective effect on the development of type 2 diabetes. But they did it apparently through different mechanisms. Mediterranean diet increased Roseburia genus, and the low-fat, high-complex carbohydrate diet increased a different bacteria. Another small study, 20 obese patients, 19 lean subjects. Obese patients had a decrease in bacterial biodiversity. Subjects with the highest gut microbial diversity had changes that you could see on CAT scan in the hypothalamus, hippocampus, and caudate nucleus. And subjects with greater abundance of Actinobacter had better motor speed and attention. Bariatric surgery affects the gut microbiome composition. It's different in patients after gastric bypass compared to obese controls. E. coli, Klebsiella, Pseudomonas are all more common. And here's the interesting part. Stool from gastric bypass patients transferred to germ-free mice. Mice had improved fat oxidation. Microbe-transplanted mice gained 43% less body fat compared to the same experiment done in mice with a control. The control was stool from gastroplasty patients, not gastric bypass. What happens in pregnancy? Women gain weight, and then some of them have enormous difficulty in shedding those extra pounds. So the gut microbiota changes dramatically from the first to the third trimester. Vast expansion of diversity between mothers, overall increase in certain bacteria, and what's called reduced richness. But when transferred to germ-free mice, third trimester microbiota induced greater adiposity and insulin resistance compared to when transferred from first trimester into germ-free mice. And this is shown schematically here. First trimester, normal microbiota, transplant it into a mouse, it remains normal. Then there's weight gain, insulin desensitization, third trimester, altered microbiota, transplant third trimester microbiota, and the mouse gets very fat and develops insulin desensitization. So long-term infant outcomes are influenced by the mode of delivery. Children born by C-section are more likely to develop type 1 diabetes, celiac disease, hospitalization for gastroenteritis, asthma, and allergic rhinitis. Now, what is this condition? You should be able to tell. Pediatric gastroenterologists in particular, right? You know it. What are you seeing? Pneumatosis, air in the portal vein, and then at surgery, necrotic bowel. So this is necrotizing enterocolitis. Study of 1,000 premature newborns, pretty horrific incidence, 6%. And you know, with ICUs and ID consultants and these magical antibiotics, we have not made a dent in the mortality. Horrific, 36% mortality rate. In our country, necrotizing enterocolitis. So it turns out premature newborns who survive the first 2 weeks have a much higher risk of dying from necrotizing enterocolitis. And the gut microbiota in these newborns are different. But what's interesting is that this dysbiosis is observed several days before the clinical event. And this may offer us a window where we can intervene now with antibiotics, maybe antibiotics and probiotics. So randomized controlled trials are occurring now in our country to see if this will make a difference. So potential window for intervention. Is there a gut-brain connection? When you say, I have a gut feeling, who's talking? Is it the gut microbiome? The immune system is active in myelin destruction. Alteration in gut flora in mice leads to an MS-like disease. It's called experimental autoimmune encephalomyelitis. And preliminary work suggests that the gut in MS patients contain bugs that drive inflammation and are low in the types of bacteria that control inflammation. Here's a reference in PNAS. Ingestion of lactobacillus strain regulates emotional behavior and central GABA receptor expression in a mouse via the vagus nerve. The vagus nerve is turning out to be very central in all this communication between the gut and the CNS and the brain. So there's crosstalk between gut microbiota and brain function. In germ-free mice, if you restrain them, it leads to an exaggerated ACTH response. But you can partially reverse this response simply by colonization with fecal material from control mice. Normal mice subjected to psychosocial stress have a decrease in bacteroides, increase in Clostridia in the cecum. In humans, probiotics modulate brain activity. Bariatric surgery has now been shown to be followed by profound changes in microbiota, but also improvement in memory, and not seen if you have gastroplasty. So this is the gut-brain axis, a complicated slide. 5-hydroxytryptamine, cytokines, bacterial molecules, fatty acids, GABA, 5-HT precursors. But I want you to pay attention to the vagus nerve and the centrality of the vagus nerve. So it turns out in Parkinson's disease, one of the earliest symptoms that people develop, years before they have neurological symptoms of tremor and mask-like facies, is constipation. So one of the questions asked is, what happens if you do a vagotomy? And somebody actually looked this up and published a paper. People who've undergone a truncal vagotomy have a lower risk of developing Parkinson's. But if they have a supraselective or parietal cell vagotomy, simply denerving the parietal cells, doesn't make a difference. Has to be truncal vagotomy. So the gut microbiota profile is significantly altered with brain microstructure and function, and this was published a few years ago. What about stroke? They took 2 colonies of mice. Group A, the mice were resistant to antibiotics. Group B, they were vulnerable to antibiotics. So they produced an ischemic stroke by simply occluding the carotid arteries. But the brain damage was 60% smaller in group B mice, the ones that were susceptible to antibiotics. Fecal transplant from mice that had reduced brain damage, now given to naive mice, they alter their gut microbiome, not been given antibiotics, right? Just given fecal transplant. Now you do the same experiment. Occlude the cerebral arteries, leading to ischemic stroke, against significant protection against brain damage seen. Composition of immune cells, good regulatory T cells, and bad gamma delta T cells altered. So we think of stroke as being ischemic, there's some edema, but there's also inflammation, cytokines, and the gut microbacteria modulate that. So it's too early, of course, to experiment in human beings, but here's a study published in Nature Medicine. Commensal microbiota affect ischemic stroke outcome by regulating intestinal immune cells. Caveat, again, this is a study in mice. What about the diet and microbe-morbid union? How many of you have heard of TMAO? Or how many of you are sending a TMAO level in your patients? Very few. When I've asked this question, virtually nobody. Doesn't matter whether it's in Santa Fe or in Boston or Cleveland, if I'm giving this talk. So it turns out trimethylamine oxide, when we consume high-fat foods, phosphatidylcholine is converted into choline, which is converted into TMA in the gut by the microbiome. And then TMA is converted into TMAO in the liver. And there's a correlation between atherosclerosis, risk of heart disease, and stroke. The correlation is regardless, irrelevant, what the CRP is, what the cholesterol is. And we all have patients who develop heart disease and stroke and have normal cholesterols. So TMAO is produced when intestinal bacteria digest the nutrient lecithin. In human subjects, give them 2 hard-boiled eggs, capsule of labeled lecithin, they'll get an increase in TMAO. But when the same subjects are given broad-spectrum antibiotics, TMAO levels are suppressed. High TMAO blood levels are associated with high risk of heart attack or stroke, independent of other risk factors and other blood test results. So Cleveland Clinic published a lead article in the New England Journal of Medicine about TMAO and risk of atherosclerosis, heart attacks, and strokes. They have a patent. For a few dollars, you can get a TMAO level checked at Cleveland Clinic. So dietary meat is a major source of TMAO. As I mentioned earlier, TMAO levels, independent risk factor for atherosclerosis. There's a very interesting animal model in mice, ApoE deficient mice. They develop robust atherosclerosis, high atheroma burden. When ApoE deficient mice are treated with antibiotics, significant decrease in atheroma burden. A high-fat Atkins diet for 4 weeks shown to be associated with high levels of TMAO, but a low-fat Ornish diet for 4 weeks associated with the low levels of TMAO. The one diet that's been shown to reverse atherosclerosis angiographically is a vegan diet. And again, it has an effect on TMAO levels. So diet indeed is destiny. Fate is shaped by genome and microbiome. This is a reference in the New England Journal of Medicine from the Cleveland Clinic, intestinal microbial metabolism of phosphatidylcholine and cardiovascular risk. Now, we all know of patients like this. They go get a double cheeseburger, double French fries, 2 apple crisps, right? 1440 calories. What drink do they order with this meal? Diet Coke, right? New total, 1441 calories. But why am I bringing this up? The worst thing our patients can do is drink diet drinks. I tell them if you want to drink water, drink coffee. You know, if you want to have a Coke or a ginger ale, have a little bit, throw the rest, but don't have diet drinks. Not sure about some of the new natural sugars like stevia and so on. So artificial sweeteners make glucose intolerance worse by altering the gut microbiota, shown in this slide. So get rid of artificial sweeteners. No more diet drinks at the Northern California GI Society annual meeting. Get rid of it. So preliminary work shows vegan diet, high fiber diet, highly fermented foods, kimchi, colostrum, all have a favorable influence on the composition of the gut microbiome. Now look at this. This is fascinating to me. Again, small studies. Autism. Pregnant mice injected with artificially created virus-like DNA, they develop an autism-like disorder. These offspring display less socialization, greater sense of being startled by sounds, fewer vocalizations. And the serum of these ASD mice contain more than 45 times the amount of 4-ethylphenyl sulfate, which is a metabolite of gut bacteria. What about kids with autism? Children with ASD have high concentrations of a similar compound, p-cresol, in their urine. Healthy mice injected with 4-EPS have a leaky gut and display ASD symptoms. Probiotic treatment with B. fragilis in ASD mice restores intestinal permeability, and 4-EPS levels return to normal. I'm convinced it's a hypothesis that when we have patients with IBD, and some of them develop erythema nodosum, pyoderma gangrenosum, sclerosing cholangitis, what's going on? I think they have an altered microbiome. It's a fruitful area of research. I tell all the residents, so what field are you going in? Neurology, rheumatology, GI, endocrine. You want to do clinical work? Yeah. You going to do basic science work? Yeah. I said, study everything about the microbiome in your discipline and do research. You will have a fruitful career. So look at this. It's a really tantalizing small study, only 18 participants, but they gave them a stool transplant. And 2 years later, most had still improvement in GI symptoms, even more. Autism-related symptoms improved even more. And the gut microbiota remained altered 2 years later. It didn't revert back to the original. So now a randomized placebo-controlled trial is warranted, and it's going to happen. There's a psychiatrist in Ireland. He's coined the term psychobiome. He's written a book called The Psychobiotic Revolution: Mood, Food, and the New Science of the Gut-Brain Connection. So there's going to be much more funding about all this research. There's a student who went to a faculty member at MIT and said, let's start a nonprofit company. They called it Open Biome. They have about 10,000 rigorously tested samples of stool, negative for HIV, HBV, HCV, and clinical investigators from anywhere in the world can apply. They have a team board that looks at the protocol. If they approve, they ship them samples for free. There's a guy living in Seattle, Naveen Jain. He has started a new company called vBiome. And he's got people like Urban Meyer at UCLA, who's a professor, who's written a book, has a blog about the microbiome on his board. So gut microbes are essential. They're not freeloaders. They digest food. They produce anti-inflammatory chemicals and compounds. They guide the immune system to distinguish friend from foe. A word about prebiotics and probiotics. Prebiotics are selectively fermented products that confer changes in the composition and/or activity of the GI tract microflora, and we think confer health benefits. But probiotics are ingested microorganisms that are associated at the moment with limited proven benefits, right? Antibiotic-associated diarrhea, pouchitis. There's one study in gastroenterology, hepatic encephalopathy from Chandigarh, India, VSL3 probiotic, marked improvement, child's pew classification, less hospitalization for recurrent hepatic encephalopathy, needs to be duplicated. So very few, yet probiotics, global sales in 2018, 40 billion. And in next year, estimated to reach 64 billion. I have so many friends and physicians who will turn to me and say, Sanjiv, which probiotic should I take? I said, I have no idea. I think in the future, we'll study your stool and we'll say, for you, Jim, you should take VSL3, Philips Colon Health, and have Greek yogurt twice a week. And then we'll check your microbiome a month later and see if it made a difference. But there are companies that are making a profit right now on the internet. You submit a stool, they'll charge you $250, and they'll tell you what 8 things to do, what food to eat, whether to drink coffee, whether to take a probiotic. So one of the challenges regarding probiotics, by definition, supposed to contain 100 million to a billion, sometimes even 10 billion bacteria. But there's no quality control. And if they say it contains, 500 million of this particular bacteria, you and I have no idea what we're taking. There's also no good standardization of the stool analysis. I happened to be listening to a BBC talk, and this correspondent submitted her stool to a well-reputed lab at Oxford, and then another lab in London. Same sample of stool, totally divergent analysis of the stool. So standardization, cost, novel ways of delivery. There are people who are combining bacteria with other drinks, including chocolate, to see if it would be a successful combination. So more than a century ago, Elie Metchnikoff, also a Nobel laureate, said the dependence of the intestinal microbes in the food makes it possible to adopt measures to modify the flora in our bodies and to replace the harmful microbes by useful microbes. What about therapeutic complications? We all know in GI that for refractory C. diff, the best treatment is not vancomycin, it's stool transplant, right? So you all saw this article some years ago. This was a clever study done by the Mass General. They had 20 patients with recurrent, difficult to eradicate C. diff. They got a stool transplant. Clever part, no NG tube, no colonoscopy. Took the stool, froze it, put it in a capsule, tested it a month later, negative for HIV, HBV, HCV, told the patient, pinch your nose, swallow it. All right, 14 of 20, one treatment, resolved. 4 of 6 non-responders, diarrhea resolved with retreatment. No adverse effects. I call it the frozen poopsicle. If stool transplant is successful in refractory C. diff, we can say the number 1 treatment for refractory C. diff is number 2. Okay, this is the study I mentioned earlier about probiotics beneficial in hepatic encephalopathy. Marked improvement in child's pew and MELD scores, published in gastroenterology. Needs to be validated. Now look at this. Probiotics taken during pregnancy influence maternal and child outcomes. Probiotic milk consumption during early pregnancy reduces the risk of premature death, but probiotic milk consumption during late pregnancy reduces the risk of preeclampsia, which is a pretty horrific disease. Sometimes we see HELLP syndrome, together with it, hepatic rupture, rare occasion, and so on. We have to be concerned about stool transplant. The FDA has recently put a ban just a few weeks ago. You know about that? There were 2 patients in our country who were immunodeficient and got a stool transplant from a healthy donor, and they developed multi-drug-resistant E. coli, sepsis, and 1 died. They tested the donor, perfectly healthy, had the same bug, exact same bug. So they transmitted it. So we have to be very careful about this. Now, there's been a sharp rise in rate of C-section in recent years, accompanying increase in asthma, allergies, eczema, obesity. Is the microbiome to blame? We don't know, right? The women who undergo C-section also, there are many other factors. They may often receive antibiotics routinely after C-section. They have a greater prevalence of obesity. They have lower rates of breast milk feeding. So the FDA has now approved 2 large-scale studies, one in Virginia, one in New York, up to 800 patients randomized, where half of them will get the swab from the vagina and the mouth of the newborn on the skin, and the other half will get a placebo. I'm calling this bacterial baptism. Okay, what about microbiome and longevity? The fruit fly has remarkably similar to mammals in terms of biochemical pathways. Now, fruit flies fed with a synbiotic. Synbiotic is a probiotic combined with a herbal supplement, had reduced inflammation, reduced oxidative stress, and a 60% increase in longevity. Pretty remarkable. Probiotics dramatically change gut microbiota composition and in respect to how we even metabolize food. So we don't know if this applies to human beings. In human beings now, the studies that are being done are caloric deprivation, consuming less than 600 calories, 400 calories. They look very gaunt. They look miserable to me when I see their pictures. Maybe metformin may have, somebody mentioned that earlier in a talk, have an effect on longevity. Longer telomeres, Elizabeth Blackburn got the Nobel Prize 2009, describing telomeres. Who has shortened telomeres? Mothers of chronically severely depressed, disabled children. The stress day in, day out. Caregivers of people with Alzheimer's. Who has longer telomeres? People on the Mediterranean diet, people who exercise, people who drink coffee, and people who meditate, and maybe people on metformin. So we'll get the answers in the next decade. This is Open Biome. I mentioned this. They've already shipped more than 30,000 samples to a network of almost 1,000 hospitals around the world. So let's summarize. Microbes appeared 3.5 billion years ago. There's crosstalk. They've been doing it for billions of years. Gut microbiota affect obesity, cardiovascular disease, neurological disease, arthritis, necrotizing enterocolitis, diabetes, colon cancer, I didn't mention IBS, IBD, pregnancy outcomes, longevity, even our response to chemotherapy, modulated by the microbiome. Side effects of chemotherapy, modulated by the microbiome. So it's pretty impressive. Composition of the microbiota is in flux, altered by a diverse array of factors. And right now, the therapies are limited: C. diff, hepatic encephalopathy, maybe more like autism down the road, and other chronic disabling disorders. Now, I'll conclude by asking you, have you heard of biological dark matter? Do you listen to TED Talks? Do you give TED Talks? You should do both. So I happened to listen to this TED Talk by Nathan Wolfe. He's an explorer, and he's a biologist. And he gave a talk to kids in school, and a 13-year-old girl stood up at the end during the Q&A, and she said, "What's the next frontier in science for human beings to conquer?" And it got him thinking. And then he developed this brilliant TED Talk, and he talks about biological dark matter. So it is uncategorized genetic material found in humans that does not fall under the 3 existing domains of life. It may well be a 4th domain of life, yet to be fully understood. And here's what's fascinating. It accounts for 40 to 50% of the genetic material in the human gut, 20% of the genetic material in the nose, and up to 2% of genetic material in sterile blood. We have no idea what it's doing, but we're transfusing it into people, and it's in our gut. So Nathan Wolfe gives this talk, What's Left to Explore. He gave this TED Talk in 2015, really worth, just Google Nathan Wolfe TED Talk, and you'll hear it, brilliant. So we're all familiar with this saying and this picture. We've only seen the tip of the iceberg. But an African colleague of mine came up to me about 6 months ago, and he said, "Sanjiv, we use a different way of saying this." He said, "We say, we've only seen the head of the hippo. Thank you very much for your attention.